Welcome to AI Paper Podcasts. We're the show that digs into the latest research so you don't have to get lost in pages and pages of methods and appendices.
Yep. We try to pull out the key stuff for you.
And today, we are tackling something I think is a really fundamental problem in, well, any computational science, really. It's that gap between a published paper and actually using the methods described.
The friction.
Exactly. We're unpacking a paper from Stanford researchers called Paper2Agent, and the promise here is pretty bold, converting that static, passive scientific paper into an active, interactive AI agent you can talk to.
It sounds almost like science fiction, but it's quite practical the way they've approached it.
Our mission today, then, is to really understand the system they built, this architecture that makes it possible. The idea is basically turning every research paper into its own little personalized, executable API.
Yeah, moving beyond just reading about a method to actually interacting with it instantly.
Okay, so for those short on time, let's do the 1-minute rundown. What are the absolute key takeaways here?
All right, 1 minute. So the core problem. Traditional research papers, especially the really computational ones like, say, AlphaGenome, which we'll get into, they're passive.
Right, just text and figures.
And they're often locked behind these huge technical barriers. Complex dependencies, setting up the right environment, maybe hours of debugging just to run one line of code. It makes sharing and reusing methods really slow.
So the solution proposed here is Paper2Agent. It's this automated framework, uses a multi-agent system to turn the paper and its code into these reliable, interactive AI agents.
And the mechanism, the how, is built around something called the Model Context Protocol, or MCP. Think of it like a standard way for the scientific method in the paper to expose itself and to make itself understandable to LLMs like Claude Code.
So it structures the paper's contributions into tools the AI can use.
Exactly. Executable tools accessible via natural language. Just ask it a question.
And the outcome. Because this sounds great in theory.
Well, the results they show are pretty impressive. The agents they created achieved 100% accuracy in reproducing the original results in their case studies.
Okay, that's crucial for validation.
But more than that, they could execute complex, totally new scientific queries that weren't in the original paper. So it's not just about replication, it's like a new way to adopt methods and collaborate almost immediately.
That's the paradigm shift bit.
Yeah, I think what's really fascinating is how it turns a document encoding knowledge into an entity that is knowledgeable and can execute things. For users, especially maybe non-experts in coding, it just radically lowers that technical barrier.
Like the biologist example you mentioned, if you need genomic data insights, you maybe don't need a PhD in Python anymore just to access the tool.
Precisely. You talk to the agent.
Okay, let's unpack this more slowly now, starting with that pain point. I think everyone listening who works in computational research knows this feeling intimately. The paper is the standard unit, right?
Yeah.
But it's passive.
Totally passive.
And we all know that ritual. You read the paper, it looks amazing, you click the GitHub link.
Then the fun begins.
Yeah. Hours fighting with conda, pip, different versions, operating systems, just trying to get the author's demo code to run on your machine. It's often non-trivial, to put it mildly.
It really is the ultimate friction point. And the paper uses a great example, AlphaGenome. It's this powerful genome scale model. Fantastic science.
Right.
But if you're, say, a bench biologist, the person who most needs the biological interpretation it offers, you're probably siloed off. You need pretty specialized knowledge just to deploy it. Installing the specific environment, importing the right modules, handling API keys, understanding how the API itself is structured.
So the core science is just sitting there, kind of locked away, unusable for a whole chunk of the potential audience. That's a massive bottleneck for scientific progress, isn't it?
It absolutely is. And that's why they argue AI agents are kind of the necessary next step. These are systems, powered by LLMs, connected to tools and APIs.
They don't just read instructions.
Right. They can reason, plan multi-step actions, bring in context. And because you interact using natural language like English, it just cuts through all those technical setup hurdles.
You just talk to the method. Okay, but reliability is key. How does Paper2Agent actually make this transition from paper to usable agent reliable? This gets us to the core architecture, right?
Yeah.
Centered around this MCP, the Model Context Protocol.
Yeah, think of the MCP as like a universal translator for scientific methods. Paper2Agent takes the paper and its code and builds an MCP server. This protocol basically forces the paper's methodology into a standardized structure.
Ah, okay. So it imposes order. It creates structured APIs and tools that the LLMs can directly understand and use.
Exactly. Chaos standardized.
So the paper effectively becomes this local server, speaking a language the agent layer can understand. What are the main pieces inside that server?
There are three main things that live inside this MCP server. First up, you got the MCP tools. These are the actual executable functions. They capture the core methods from the paper. So for Alpha Genome, an MCP tool might be the function that takes a genetic variant you give it and then spits out the predictions and maybe some visualizations.
And
Crucially, these tools come bundled with their own pre-configured, containerized environment.
Got it. So dependency hell bypassed immediately. The tool is the environment. What's number two?
Number two is MCP Resources. These are all the static assets from the paper, but standardized and stored so the agent can easily find and query them.
So like the manuscript PDF itself?
Yeah, the text, the original code repository, supplementary data tables, figures, links to datasets used for training, anything static the agent might need to refer back to.
Okay, tools and resources. What's the third component? Sounds like it might be about workflow.
You got it. That's the MCP prompts. These are basically concise instructions or recipes that guide the agent through complex, multi-step scientific workflows. These are often inferred from the paper's tutorials or the code base structure.
So for something like Scanpy, which we'll discuss later, it wouldn't just be cluster this data.
Exactly. The MCP prompt would encode the sequence, first do quality control, then normalize, then select features, then reduce dimensions, then cluster. It knows the right order.
Right, because order matters hugely in bioinformatics. Now, reliability. We're talking about LLMs interacting with code. The risk of code hallucination, the LLM just making up plausible but wrong code feels very real. How do they ensure these tools are actually trustworthy?
That's a critical point. They have this rigorous validation step they call locking the tools.
Locking.
Yeah. So after a potential tool is generated, it's automatically run and validated against the results or figures reported in the original paper's tutorial, using the exact data from that tutorial.
Ah, okay. Direct comparison.
If it doesn't reproduce that original output perfectly, it's considered unlocked and isn't included in the final MCP server. It has to work exactly as advertised in the source material.
That's a strong safeguard. And transparency.
For transparency, every locked tool also contains a traceable reference, like a link back to the specific part of the original source code, usually on GitHub, that it represents.
Okay, so it's not just generating black box code. It's generating validated, traceable, essentially production-ready functions based on the original work. Now, the automation itself, how does this conversion happen? It's not just one big AI reading a PDF, is it?
No, it's actually a multi-agent system doing the work.
Yeah.
Implemented using Claude code, apparently, and coordinated by a main orchestrator agent. Think of it like a little digital lab team with specialized roles.
Okay, who's on the team? Who kicks things off?
Well, first you need a clean lab bench, right? So there's the environment manager agent.
It's a digital custodian.
Ha, exactly. Its job is to set up a clean, reproducible workspace, like a virtual environment or a container, and handle installing all the dependencies listed in the paper. Make sure everything plays nicely together.
Okay, environment sorted. Then who figures out the actual science bits?
That's the job of the extraction agent. It uses this specialized module. They call it the Tutorial Tool Extractor Implementer. Quite a mouthful.
Rolls right off the tongue.
Doesn't it? But its job is basically to read the code and the tutorials, identify the key functions or steps that represent the core methods, and then convert those into reusable, parameterized functions ready to become MCP tools.
And then the crucial validation step we just talked about, the locking.
Right. That's handled by the testing agent using another module called the Test Verifier Improver. This agent takes the candidate tool from the extraction agent, runs it using the data from the tutorial.
Okay, wait, you mentioned it only uses the tutorial examples for testing. Doesn't that create a risk? Like maybe we're just validating that the tool works for the cherry-picked examples the authors put in the tutorial, not that it's truly generalizable.
That's a really fair question. The initial locking process does rely on the tutorial examples, primarily to confirm basic reproducibility and check that the environment's stable. It's like the first sanity check. But the real test of generalizability comes later, when the final agent layer, the one the user interacts with, tries to use these locked tools to answer novel questions that go beyond the tutorial scope.
Ah, I see.
If the tool was brittle, if it only worked for that specific tutorial data, the agent's attempt to use it for a new task would likely fail or give garbage results. So the system implicitly favors the creation of robust generalized functions during the extraction phase because those are the ones that will actually be useful later on.
That makes sense. The proof is in the pudding when you try to use it for something new. Okay, let's see the payoff then. Let's dive into that Alpha Genome case study. Remind us what AlphaGenome does.
Right, AlphaGenome is an AI model that predicts the functional impact of single nucleotide variants. Tiny changes in DNA on how genes are regulated. Really cutting-edge genomics.
And Paper2Agent tackled this. How did it go?
The automation speed was pretty remarkable. The system generated 22 distinct AlphaGenome MCP tools. These covered things like scoring variant effects, predicting impacts at the sequence level, and generating various visualizations functions like score variant effect.
And the time taken?
About 3 hours, running entirely on a standard personal laptop, with zero human intervention needed during that process.
3 hours from paper and code to a suite of 22 validated tools. That's impressive. What about accuracy? Did the agent actually work?
Yes. The results were frankly spectacular. They tested the resulting AlphaGenome agent on 2 sets of queries and achieved 100% accuracy on both.
Wow. Okay, what were the 2 sets?
The first set was 15 queries taken directly from the original AlphaGenome tutorial. This basically confirmed that the agent could perfectly reproduce the paper's own examples. Basic check passed.
Okay, reproducibility confirmed. What was the second set? This is the generalizability test you mentioned.
Exactly. The second set consisted of 15 novel queries. These involved genetic positions, specific allelic changes, and different cell type contexts that were never mentioned or tested in the original paper or its tutorials.
And it got 100% on those two.
100% accuracy.
Yeah.
This is the key result, I think. It shows the agent didn't just memorize the tutorial. It successfully internalized the underlying methodology and could apply it correctly to new, unseen scenarios.
That really does demonstrate robustness. They included a more complex example too, right? Interpreting a specific GWAR's hit.
Yes, the LDL cholesterol-associated variant, HR1.109274968.GT. This was a great showcase. The agent didn't just run one tool, it used multiple planning action observation cycles. It queried different tools, analyzed the results, and then generated a full report interpreting the variant's likely impact, citing the model's evidence from the tools it ran.
And what was the interesting finding there?
Well, this is where the agent kind of transitioned from just being a tool runner to acting more like a scientific collaborator. In its generated report, the agent really highlighted the gene SRT1. Yeah, it cited a very high quantile score for the variance effect near SRT1 in liver tissue, which makes biological sense because SRT1 is involved in VLDL secretion related to cholesterol.
Okay. But wait, I think I remember the original AlphaGenome paper. Didn't they focus more on 2 other genes at that same locus, CLSR2 and PSRC1, when discussing that variant?
You remembered correctly. The original paper did emphasize CLSR2 and PSRC1. So the agent, based purely on the quantitative evidence it pulled from the model via the MCP tools, actually prioritized a different gene than the original authors did in their narrative discussion.
So the agent effectively challenged the author's interpretation based on their own model's data.
In a way, yes. Now, to be clear, they did a manual check using the GTX database, and it turns out all 3 genes, SR1, CLSR2, and PSRC1, have significant expression QTLs associated with that variant. So biologically, all 3 are potentially relevant.
But the agent's interpretation differed based on the model scores.
Precisely. It highlights a really valuable aspect of these agents. They enable this dynamic reassessment of hypotheses using the model's evidence in real time, potentially uncovering insights or interpretations that go beyond the specific narrative or selected examples presented in the original static paper.
That is genuinely profound. The agent isn't just reproducing a table, it's performing active scientific reasoning and interpretation based on the underlying model.
Okay, incredible. So we've seen it handle complex genomics. What about another common challenge? Applying a new method to totally new, messy data sets. That brings us to the tissue study.
Right, tissue, which stands for Uncertainty-Aware Spatial Transcriptomics Analysis. This case study focused exactly on that pain point. You read a cool new methods paper, and now you want to try it on your data, or maybe some other public data, quickly.
How did the agent help here? The Paper2Agent system generated 6 MCP tools for tissue, covering things like running the core prediction model, constructing uncertainty intervals, and performing downstream analyses that account for that uncertainty. But beyond just the tools, the agent also acted as an interactive guide.
How so?
It could provide structured explanations, like through a Q&A, about what inputs were needed for each tool and what kind of outputs you should expect. It helps you understand how to use the method.
And the data handling part was particularly neat here, wasn't it? Using the MCP resources.
Absolutely. This was clever. The system parsed the data availability section of the tissue paper and turned it into a structured, machine-readable data registry within the MCP resources.
So instead of just text saying data is on Zenodo, it created a table.
Essentially, yes. listing things like species, tissue type, data modality, and the direct download URL, often via the Zenodo REST API.
And the benefit.
The agent could then use this structured registry. You could literally ask the agent, find the mouse brain spatial transcriptomics dataset mentioned in the paper, download it, and run the tissue prediction. And it could do that, automatically filter the registry, fetch the data, and run the tool.
Wow. That just removes hours, maybe days of manual searching, downloading, and formatting that usually precedes applying a method to new data. Okay, one more case study. Scanpy. This is different again, right? It's not a single model, but a huge, widely used software package for single-cell analysis.
Exactly. Scanpy is massive and modular. Trying to agentify the entire thing might be overkill or even nonsensical. So Paper2Agent took a pragmatic approach.
What did it do?
It focused on generating tools for the most common standardized workflow that people use Scanpy for: pre-processing and basic clustering. It created 7 core tools covering steps like quality control, normalize data, find highly variable genes, and so on.
So targeting the 80% use case.
Pretty much. It's perfect for the user who just needs that standard pipeline run reliably without needing to become an expert in every obscure Scanpy function.
And this is where those MCP prompts become really critical for orchestration, right? Because in single-cell analysis, the sequence of these steps is absolutely vital.
Precisely. You can't normalize counts before you've done quality control filtering, for example. Just doesn't make sense. The MCP prompts encode this standard pipeline QC, normalization, feature selection, dimensionality reduction, clustering, maybe even basic cell type annotation in the correct locked-in sequence.
So the user doesn't have to manually call tool 1, then tool 2, then tool 3 in the right order?
Nope. The user basically just needs to provide the path to their input data file. The agent, guided by the MCP prompt, automatically sequences and executes the entire workflow using the appropriate MCP tools in the correct order. It takes away that cognitive load and potential for error in sequencing the steps.
And did they validate this? Did the Scanpy agent reproduce known results?
They did. They tested it on publicly available 10x Genomics PBMC datasets, a standard benchmark, and confirmed that the agent successfully reproduced the key steps of the workflow and yielded results consistent with standard human-executed analyses you'd find in published studies.
Okay, so across these diverse case studies, a deep learning model, a stats method with complex data needs, a modular software toolkit, Paper2Agent seems to deliver on its promise.
Yeah, if we try to tie this all together, pull back to the big picture, Paper2Agent really does seem to represent a shift. It takes research from being just a passive artifact, something you read, maybe struggle to replicate,
into an active, almost collaborative entity, this knowledgeable entity you mentioned earlier.
Exactly. It bridges that gap between publishing a finding and allowing others to practically apply and build upon it, potentially much faster and more reliably than before.
It feels like more than just a technical optimization. It changes the nature of scientific communication, doesn't it? What could this mean for things like scientific rigor or even future publication standards?
I think it has significant implications there. The very ease with which a paper can be agentified by a system like Paper2Agent could become a practical, maybe even objective, measure of its reproducibility and rigor.
How so?
Well, think about it. If the original code is poorly documented, full of bugs, not modular, uses hard-coded paths, the Paper2Agent system will likely struggle or fail entirely to extract reliable, locked tools.
Right. Garbage in, garbage out, even for the agent builder.
Exactly. So authors who structure their code and document their methods clearly, making them inherently machine-readable and modular, will produce papers that are much easier to turn into reliable agents. It incentivizes good practice.
I can absolutely see journals, especially top-tier ones, starting to ask for this. Maybe not tomorrow, but soon.
I agree. It's not hard to imagine an agent availability section becoming standard in publications. Specifying how the paper's core contributions have been embodied as an interactive agent, maybe downloadable or accessible via a standard API.
That would really push authors to design for machine interactivity from the start, which, as you say, benefits the whole scientific community.
Absolutely.
So wrapping up, we've seen how Paper2Agent can turn static papers into active tools, boosting reproducibility and accessibility. What's the final sort of provocative thought here? Where could this lead?
Well, it raises a fascinating, maybe slightly futuristic question.
Yeah.
Once we have scientific knowledge reliably encoded in these active executable agent entities, the potential might extend beyond just humans interacting with individual paper agents.
You mean agents interacting with other agents?
Exactly. What scientific breakthroughs become possible when these agents can collaborate? Imagine a newly published agent representing a cutting-edge analytical method, automatically discovering and interacting with another new agent representing a freshly released dataset relevant to its domain. You could potentially have this dynamic, interoperable layer of scientific intelligence forming, constantly integrating new knowledge and capabilities, potentially driving discovery at a speed limited by computation, not by the friction of human integration and translation. What happens when research isn't just defined by what it describes, but by the interactive, potentially collaborative entities it creates?
That's a huge thought to end on. An ecosystem of scientific agents. Definitely something to mull over.